Properties of food substances. Food substances can have a variety of properties, including 1. Nutritional content. This includes macronutrients, carbohydrates, fats, and proteins, and micronutrients, vitamins and minerals. 2. Taste. Food substances can be sweet, savory, sour, salty, or bitter. 3. Texture. This can include the degree of crispiness, chewiness, or juiciness of a food. 4. Color. Foods can range in color from white to black, and can include various shades of red, orange, yellow, green, and blue. 5. Odor. The aroma of food can be a defining characteristic of its flavor. 6. Shelf life. Food substances can have different expiration dates or storage requirements based on their composition and packaging. 7. Allergenic properties. Some food substances can cause allergic reactions in some people. 8. Addictive properties. Certain foods, such as those high in sugar, salt, or fat, can be addictive to some people. These are just a few examples of the many properties of food substances that contribute to their taste, nutritional value, and overall desirability. 1. Properties of carbohydrates. Properties of monosaccharides. Properties of monosaccharides, which are simple sugars, include 1. Sweetness. Most monosaccharides are sweet to taste. 2. Solubility. Monosaccharides are soluble in water and other polar solvents. 3. Crystallinity. Most monosaccharides have a crystalline form. 4. Reducing properties. Monosaccharides have reducing properties due to the presence of a free carbonyl group. 5. Reactivity. Monosaccharides can react with other compounds to form glycosides and other complex carbohydrates. 6. Isomerism. Monosaccharides occur in different isomeric forms, such as glucose and fructose. 7. Configurational isomers. Monosaccharides can have D isomers or L isomers depending on the position of hydroxyl groups around the chiral carbon. These are some properties specific to monosaccharides, and they can vary depending on the specific type of monosaccharide. B. Properties of disaccharides. Properties of disaccharides, which are formed by the condensation of two monosaccharides, include 1. Sweetness. Many disaccharides are sweet, such as sucrose and lactose. 2. Solubility. Disaccharides are soluble in water and other polar solvents. 3. Reducing properties. Disaccharides have reducing properties due to the presence of a free carbonyl group in one of the monosaccharide units. However, some disaccharides may not have reducing properties due to their structure. 4. Hydrolysis. Disaccharides can be hydrolyzed by enzymes or acid to form the component monosaccharides. 5. Melting point. Disaccharides tend to have a higher melting point than monosaccharides due to their larger size. 6. Reactivity. Disaccharides can react with other compounds to form more complex carbohydrates. 7. Isomerism. Some disaccharides, such as lactose, have isomeric forms depending on the orientation of the glycosidic bond. These are some properties specific to disaccharides, and they can also vary depending on the specific type of disaccharide. C. Properties of polysaccharides. Properties of polysaccharides, which are composed of many monosaccharide units, include 1. Insolubility. Many polysaccharides are insoluble in water and other solvents due to their large size and complex structures. 2. Functionality. Polysaccharides have many different functions, including energy storage, like starch and glycogen, structural support, like cellulose, and cell signaling like glycosaminoglycans. 3. Molecular weight. Polysaccharides have a high molecular weight due to their large size and repeat units. 4. Reactivity. Polysaccharides can react with other molecules to form glycoconjugates and play a role in cell recognition and signaling. 5. Hydrolysis. Polysaccharides can be hydrolyzed by enzymes or acid to form the component monosaccharides. 6. Biodegradability. Many polysaccharides are biodegradable and can be broken down by microorganisms in the environment. 7. Isomerism. Some polysaccharides may have isomeric forms depending on the orientation of the glycosidic bonds within the molecule. These are some properties specific to polysaccharides, and they can vary depending on the specific type of polysaccharide. 2. Properties of proteins. Properties of proteins, which are composed of amino acid monomers, include 1. Molecular weight. Proteins have a high molecular weight due to their large size and complex structures. 2. Solubility. The solubility of proteins depends on the amino acid composition and the environment, such as pH and ionic strength. 3. Shape and conformation. Proteins have a defined shape and conformation that determine their function. This shape is critical for the interactions between proteins and other molecules in the biological system. 4. Stability. Proteins have different levels of stability depending on their composition, structure, and environment. They can be denatured by changes in pH, temperature, and other environmental factors. 5. Functionality. Proteins have a wide range of functions, including catalysis, regulation, transportation, and structural support. 6. Isomerism. Proteins occur in L-amino acid isomers. 7. Reactivity. Proteins can undergo post-translational modifications, such as phosphorylation, methylation, and glycosylation, that affect their function and properties. These are some properties specific to proteins, and they can vary depending on the specific type and function of protein. 3. Properties of lipids. Properties of lipids, which are diverse molecules that are insoluble in water, include 1. Hydrophobicity. Lipids are hydrophobic, water insoluble, due to their nonpolar nature. 2. Solubility. Lipids are soluble in organic solvents such as ether, chloroform, and benzene. 3. Energy storage. Lipids are an important energy source in the body, with stored fats being the primary long-term energy reservoir. 4. Structural roles. Lipids can also play structural roles, such as in cell membranes, where they form a bilayer that separates the cell from its environment. 5. Insulation. Some lipids like adipose tissue act as an insulator to maintain body temperature. 6. Diversity. Lipids are a diverse group of molecules that include triglycerides, phospholipids, steroids, and waxes, each with their specific properties and functions. 7. Reactivity. Lipids can be oxidized to produce energy, and some undergo hydrolysis to release fatty acids or other products. These are some properties specific to lipids, and they can vary depending on the specific type and function of the lipid. Common reagents and chemicals used to determine food properties. There are various reagents and chemicals used to determine food properties, depending on the specific properties of interest. Some common ones include 1. Benedict's reagent, used to detect reducing sugars such as glucose and fructose. 2. Biuret reagent, used to detect the presence of proteins. 3. Iodine solution, used to detect the presence of starch. 4. Ethanol, used in extractions and in determining the lipid content of food. 5. Phenolphthalein, used to detect the presence of alkalinity or acidity in food. 6. Bromothymol blue, used to measure pH. 7. Fehling solution, used to detect the presence of reducing sugars. 8. Sulfuric acid, used in digestions and in determining nutrient content. These are just a few examples, and there are numerous other chemicals and reagents used in food analysis and testing, depending on the specific properties of interest. Practical Activity 1. To test for starch. Give the requirements, procedures and results. Requirements. Iodine solution. Food sample e.g., potato, rice, bread. Test tubes. Water bath or heating source. Procedures. 1. Cut a small piece of the food sample and place it into a test tube. 2. Add a few drops of iodine solution to the test tube on top of the food sample. 3. Shake the test tube gently to mix the contents. 4. Place the test tube in a water bath or heat it on a hot plate for a few minutes. 5. Observe the color change in the solution. Results. If starch is present in the food sample, the iodine solution will turn from a yellow-brown color to a dark blue-black color. This indicates that starch is present in the food sample. If no starch is present, the iodine solution will remain yellow-brown. Practical Activity 2. Aim. To test for reducing sugars. Give the requirements, procedures and results of the experiment. Requirements. Benedict's reagent. Food sample e.g., glucose solution, fruit juice. Test tubes. Water bath or heating source. Pipette or syringe. Procedures. 1. Take a small amount of the food sample and place it in a test tube. 2. Add an equal amount of Benedict's reagent to the test tube. 3. Mix the contents of the test tube thoroughly. 4. Heat the test tube in a water bath or use a heating source such as a hot plate for a few minutes. 5. Observe the color change in the solution. Results. If reducing sugars are present in the food sample, the color of the solution will change from blue to green, yellow, orange, or red-orange, depending on the concentration of the sugar. The greater the concentration of reducing sugars present, the stronger the color change. If no reducing sugars are present, the solution will remain blue. The intensity of the color change can be compared to a color chart to determine the approximate concentration of reducing sugars in the sample. Practical Activity 3. Aim. To test for non-reducing sugars. Give the requirements, procedures and results of the experiment. A practical activity to directly test for non-reducing sugars is a bit more complicated than for reducing sugars, as the non-reducing sugar first needs to be broken down into its constituent reducing sugars. Here is one way to test for non-reducing sugars by breaking them down first. Requirements: hydrochloric acid HCl, Benedict's reagent, food sample e.g., sucrose solution, test tubes, water bath or heating source, pipette or syringe. Procedures: 1. Take a small amount of the food sample and place it in a test tube. 2. Add some dilute hydrochloric acid solution to the test tube. 3. Heat the test tube in a water bath or use a heating source such as a hot plate for a few minutes to allow the hydrolysis reaction to occur. Note: hydrolysis breaks down non-reducing sugars into their constituent reducing sugars. 4. Once the hydrolysis is complete and the solution is cooled, neutralize the acid by adding sodium hydroxide, NaO, solution slowly until the solution becomes neutral. 5. Add an equal amount of Benedict's reagent to the test tube. 6. Mix the contents of the test tube thoroughly. 7. Heat the test tube in a water bath or use a heating source such as a hot plate for a few minutes. 8. Observe the color change in the solution. Results: if non-reducing sugars are present in the food sample, the hydrolysis step will break it down into its constituent reducing sugars. The reducing sugars produced will then react with the Benedict's reagent, causing the color of the solution to change from blue to green, yellow, orange, or red-orange, depending on the concentration of the reducing sugars. The greater the concentration of reducing sugars present, the stronger the color change. If no reducing sugars are present, either due to no non-reducing sugars being present initially, or due to the hydrolysis not being successful, the solution will remain blue. The intensity of the color change can be compared to a color chart to determine the approximate concentration of reducing sugars in the sample. Practical activity 4. Aim to test for fats oils. Give the requirements, procedures and results of the experiment. Requirements: food sample e.g., vegetable oil, butter, ethanol, Sudan 3 stain, test tubes, water bath or heating source, beaker or boiling tube, paper towel. Procedures: 1. Take a small amount of the food sample and dissolve it in ethanol. 2. Pour the ethanol solution into a test tube until it is about a quarter full. 3. Add a few drops of Sudan 3 to the test tube. 4. Shake the test tube to mix the contents together. 5. Heat the test tube in a water bath or using a heating source like a Bunsen burner. 6. Observe the top layer of the solution for any color change. 7. Place a drop of the solution on a piece of paper towel and observe the color of the stain. Results: if fats are present in the food sample, the Sudan 3 stain will bind to the fat molecules and cause the top layer of the solution to turn red-orange in color. The more fat molecules present, the more intense the color change will be. If no fats are present in the sample, the Sudan 3 stain will not bind to anything and the solution will remain the same color as before the Sudan 3 was added. Note: this experiment may not work for all types of fats, as some fats may not bind as well to the Sudan 3 stain as others. Additionally, the concentration of Sudan 3 and the amount of sample used must be controlled for accurate results. Practical activity 5. Aim, test for ascorbic acid, vitamin C. Give the requirements, procedures and results. Requirements: food sample e.g., fresh fruit, fruit juice, 2 test tubes, 10% TCA trichloroacetic acid, 2% 2,6-dichlorophenylindophenol, DCPIP, solution, distilled water, dropper or pipette. Procedures: 1. Take a small amount of the food sample e.g., a piece of fresh fruit, and squeeze its juice into a test tube. If using fruit juice, take a small amount and pour it into the test tube. 2. Add 2-3 milliliters of distilled water to the test tube and mix the contents. 3. Add 3-4 drops of 10% TCA solution and mix the contents. 4. Centrifuge the test tube or let it stand for a few minutes until a precipitate forms. 5. Take another test tube and add 2-3 milliliters of distilled water and 3-4 drops of 2% DCPIP solution. 6. Mix the contents of the second test tube well. 7. Add the TCA solution obtained in step 4 drop by drop into the second test tube, shaking the second test tube after every addition. 8. Continue adding the TCA solution until the blue color of DCPIP solution disappears. Results: if ascorbic acid, vitamin C, is present in the food sample, it will reduce the blue DCPIP solution by donating electrons to it, causing the blue color to fade. The more ascorbic acid is present in the sample, the less DCPIP solution will be needed to turn it from blue to clear. Note, this experiment is only a qualitative test for the presence of ascorbic acid and does not indicate the exact amount present in the sample. Further tests or measurements would be necessary to determine the amount of ascorbic acid. Practical Activity 6. Aim to test for proteins. Give the requirements, procedures and results. Requirements. Food sample e.g., egg white. Test tube. Biuret reagent. Water bath or heating source. Procedures. 1. Take a small amount of the food sample e.g., a piece of egg white, and place it into a test tube. 2. Add an equal amount of water to the test tube to dilute the sample. 3. Add a few drops of biuret reagent to the test tube. 4. Shake or mix the contents of the test tube. 5. Place the test tube in a water bath or heat the contents of the test tube using a heating source like a Bunsen burner for a few minutes. 6. Observe the color change of the solution. Results. If proteins are present in the food sample, the biuret reagent will react with the peptide bonds in the protein and form a complex that absorbs light at a different wavelength than the original solution. The solution will change from blue to violet in color. If no proteins are present in the sample, the solution will not change color and remain the same. The color intensity increases with the amount of proteins present in the sample. Note, this experiment may not work for all types of proteins, as some proteins may not react with biuret reagent. Additionally, the concentration of biuret reagent and the amount of sample used must be controlled for accurate results. Practical Manual on Food Test. Introduction. Food testing is a process of analyzing food samples to determine their nutritional content, quality, and safety. It is important to perform food tests to ensure the food is safe for consumption and meets regulatory requirements. This practical manual will provide instructions for common food tests, including tests for carbohydrates, proteins, lipids, and nutritive value. Requirements. Food sample. Test tubes. Beakers. Chemical reagents specific to the test being performed. Heating source, such as a Bunsen burner or water bath. Droppers or pipettes. pH paper or pH meter, if testing acidity or alkalinity. Balance. Procedures. 1. Test for carbohydrates. Take a small amount of the food sample and add a few drops of Benedict's reagent. Heat the mixture in a boiling water bath or with a heating source. Observe the color change of the mixture, which indicates the presence of reducing sugars. 2. Test for proteins. Take a small amount of the food sample and add a few drops of biuret reagent. Mix the contents and observe the color change of the mixture, which indicates the presence of proteins. 3. Test for lipids. Take a small amount of the food sample and add a few drops of Sudan IV solution. Mix the contents and observe the color change of the mixture. A red color indicates the presence of lipids. 4. Test for nutritive value. Weigh the food sample and record its weight. Burn the food sample in a crucible until it turns into ash. Weigh the ash and record its weight. Calculate the percentage of ash as follows: ash weight sample weight, x 100. The percentage of ash represents the amount of minerals in the food sample. Results. The results of each test can be observed by a color change or a clear indication of the presence or absence of the substance being tested. The results provide information about the nutritional value and quality of the food sample. The nutritive value test provides information about the mineral content of the food. Conclusion. Food tests are important in ensuring the safety and quality of food products. The proper handling of equipment and chemical reagents is important to obtain accurate results. The results of the tests should be compared to standards and limits set by regulatory agencies. Food manufacturers should perform regular testing to ensure their products meet quality and safety standards. Revision exercise. 1. If a solution of cooked starch is mixed with saliva and 5 samples from the mixture are tested with iodine solution at 1-minute intervals after mixing, what results would you expect if the mixture was kept at 30 degrees Celsius? If a solution of cooked starch is mixed with saliva and tested with iodine solution at 1-minute intervals after mixing at 30 degrees Celsius, the expected results would be: immediately after mixing, a blue-black color indicating the presence of starch. At 1 minute, the same blue-black color indicating the presence of starch. At 2 minutes, the color of the solution may change to a bluish or purplish color showing partial digestion of starch by the enzyme present in saliva. At 3 minutes, the color should become light purple, showing the gradual breakdown of starch with the production of maltose and smaller sugars. At 4 minutes, the color should become lighter indicating more breakdown has taken place. At 5 minutes, the color should become yellowish-brown indicating the complete breakdown of starch in the solution by the enzyme in the saliva. 2. When a food substance is boiled with Fehling solution which is blue, the mixture first turns green and finally produces an orange, yellow or red colored precipitate. This is the test for. When a food substance is boiled with Fehling solution which is blue, the mixture first turns green and finally produces an orange, yellow, or red colored precipitate. This is the test for reducing sugars, such as glucose, fructose, lactose, and maltose. The Fehling's test is a common laboratory test used to qualitatively determine the presence of reducing sugars in a given sample. The reducing sugar present in the food substance reduces the copper 2 ions in Fehling's solution to copper I ions, resulting in the formation of a colored precipitate. 3. Glucose is known as a reducing sugar because it has a free aldehyde or ketone group in its molecular structure. This functional group is capable of reducing other compounds by donating electrons and becoming oxidized in the process. In the presence of an oxidizing agent such as Benedict's or Fehling's solution, the aldehyde or ketone group of glucose donates electrons to the copper 2 ions in the test solution, reducing them to copper I ions and leading to the formation of a colored precipitate. This reaction is the basis for the detection of reducing sugars in laboratory tests. 4. A food sample which was tested by boiling with Benedict's or Fehling's solution gave an orange precipitate. This showed the food sample contained. A food sample which was tested by boiling with Benedict's or Fehling's solution and gave an orange precipitate indicates the presence of reducing sugars in the food sample. The orange precipitate is formed due to the reduction of copper 2 ions in Benedict's or Fehling's solution to copper I ions by the reducing sugar present in the sample. This reaction confirms the presence of reducing sugars, such as glucose, fructose, maltose, or lactose, in the food sample. This test is often used to detect reducing sugars in various food products and is an important test in food analysis. 5. A substance X was mixed with an enzyme and boiled with dilute hydrochloric acid, then neutralized with sodium hydrogen carbonate solution until alkaline. The mixture was heated with Benedict's solution. An orange-red precipitate was formed. What is substance X? B. Outline how colors changed from the original color to orange-red. C. Why was the temperature kept at 37 degrees Celsius? D. What is the role of hydrochloric acid in the experiment? E. Why was neutralization necessary before the addition of Benedict's solution? The substance X is most likely a reducing sugar, such as glucose or fructose. B. When the substance X is mixed with an enzyme and boiled with dilute hydrochloric acid, it is broken down into its respective monomers. After neutralization with sodium hydrogen carbonate, the mixture is heated with Benedict's solution. In the presence of reducing sugars, the blue Benedict's solution turns green, yellow, and then to an orange-red precipitate as the concentration of reducing sugar increases. C. The temperature is kept at 37 degrees Celsius to simulate the optimal temperature for the enzyme in the human body. D. Hydrochloric acid is used to break down the polysaccharides present in substance X into their respective monomers. E. Neutralization with sodium hydrogen carbonate is necessary to create a suitable pH environment for the enzymatic reaction, as hydrochloric acid is initially acidic. The acidic pH could potentially denature enzymes, affecting the results of the experiment. 6. A particular food substance is suspected to contain vitamin C. What chemical would you use to confirm the presence of vitamin? B. What are the expected results if vitamin C is present? To confirm the presence of vitamin C in the food substance, one could use 2,6-dichlorophenols, DCPIP, as a chemical indicator. B. If vitamin C is present in the food substance, it will reduce the blue DCPIP to a colorless form, indicating the presence of vitamin C. Conversely, if there is no vitamin C, the blue color of DCPIP will not change. The extent of color change can also be used to quantify the amount of vitamin C present in the sample. Therefore, a colorless solution after mixing the food substance and DCPIP would confirm the presence of vitamin C in the sample.